In this lesson, we are going to look at neurotransmitters and how they act as chemical messengers. So some basic structures before we move into some more specific structures. When we have a neuron, and we looked at that structure in our first lesson of this unit, we have all of these parts coming off of the neuron, and we would have dendrites, for example, which are going to make connections with another neuron. We also have the cell body, which contains the nucleus and the other organelles. So typically we only talk about the nucleus when we're talking about a neuron's cell body, but you have to remember there's all of the other things that have to exist within any kind of cell. And so you're still going to have your Golgi, your endoplasmic reticulum, you're going to have mitochondria, which we'll talk about in the next slide. So we have all of the other things, but textbooks typically just focus on the nucleus. And then we have this long axon, and it ends in what are known as axon terminals, which are going to make connections with another neuron, and that's going to be making a connection at the dendrite of the other neuron. Okay, so we have what's called the presynaptic neuron. So here is the presynaptic neuron, and we have the postsynaptic neuron, and that's just relative. So it's always one compared to another. So when we're looking at the blue versus what would be the pink neuron, this would be the presynaptic, and this would be the postsynaptic, because this neuron is sending information to this neuron. Versus, if we're looking at this, what would be a yellow neuron in the blue neuron, this would be the presynaptic, and this is the postsynaptic neuron, because this is sending information to this neuron. So we might even think of this as an interneuron, okay, between two different types of neurons. The space, there's a tiny space between the presynaptic and postsynaptic neuron. So between the axon terminal of one neuron and the dendrite of another neuron, and that is called a synapse. Okay, so that's the very, very small space between the two neurons. And we're going to take a close-up in the next slide. Okay, so here we have A, our presynaptic neuron, and B, our postsynaptic neuron. And so this is the axon terminal of one neuron, and this is the dendrite of another neuron. And some sort of stimulus has occurred, causing this neuron to need to send a message to this neuron. And we'll talk about how that happens a little bit in this lesson, and more so in the next lesson. And so here, some sort of information is causing this neuron to have to release what is called a neurotransmitter, which is going to travel through this synapse and bind to this neuron, so the dendrite of this neuron. And again, it's going to continue that message to where it needs to go, either a muscle or a gland, for example. So let's look at some of the structures so we can have a better understanding of what's happening. So don't forget, all of this requires energy, which is going to be created by the mitochondria. We're not going to review that right now. Spiral together in class if we need to. Next, we have what's called a synaptic vesicle. So this is a vesicle, and it is full of a neurotransmitter. And it, depending on the stimulus in the neuron, what's happening, we're going to look at all types of different neurotransmitters. In this case, let's just pretend it is acetylcholine. And acetylcholine is very much involved in the rest and digest part of the autonomic nervous system. And so this allows us to learn, develop memories, have a lot of attention, and very much involved when we're trying to learn something new or sit down and write a story or create something. So that's just acetylcholine in itself. It could be dopamine, it could be serotonin. We're going to look at different neurotransmitters. Next, we have a calcium channel. So this is very much like the channels that we've talked about previously, and it's involved in causing the release of the neurotransmitter to the synapse, and we'll look at that more in the future. Then again, we have our synaptic cleft, which is containing the synapse and just that little bit of space between the presynaptic neuron and the postsynaptic neuron. Then we have what's called a neurotransmitter receptor. So a receptor is going to be taking or binding the neurotransmitter that was released. So I've circled this one. It could be any one of these here. So, and it's labeled neurotransmitter receptor. So it's going to be binding the neurotransmitter acetylcholine that was released by this neuron. It'll bind to this receptor on this neuron. There's also autoreceptors shown here. So this released the acetylcholine, but it's also going to bind acetylcholine. And it does that in order to act as a negative feedback loop. So if all of this acetylcholine is being released and it's filling up here, and if this neuron isn't accepting it, then it's going to stop releasing acetylcholine. It's just a waste, if you will, of acetylcholine. There's also a neurotransmitter reuptake pump. So similarly, it will bind acetylcholine and take it back in to be reused. And again, it's trying to A, not waste resources, and B, it's trying to prevent overstimulation of the acceptor neuron or the postsynaptic neuron. Okay, so let's talk about a little bit of the process and what's happening. And again, a bit of an overview. We go into it more, what causes the actual release of the neurotransmitters in a future lesson. So 3 things could happen. 1, we have the reuptake of the neurotransmitter, and this is shown at A. So we either have the acetylcholine not being used, and so it's just going to get taken back up. So we have our reuptake transporter here. Or it gets used by the receptors and then released. And if it's released, again, the presynaptic neuron can take it back up and reuse it. We also have diffusion, where the neurotransmitters move across the synapse to the receptors, so shown here at B. So it's going to move along this very tiny space, obviously exaggerated in a diagram. And it's going to, again, bind to the receptor on the postsynaptic neuron, or, again, it will diffuse and be taken up by the reuptake transporter. And then lastly, we might see degradation of that neurotransmitter. So we have enzymes that are going to bind the neurotransmitter, again, acetylcholine in this case, and it will degrade it and break it down. This needs to happen so, again, we don't get this constant stimulation of this postsynaptic neuron. So recall when we have a neuron, we have a cell body and then long axon coming off of it. Along that axon, we have a constant when a message or a signal is being sent down. We have this constant changing of membrane potential along that axon. And we're going to talk a little bit today how that happens, but we go into more detail in a future lesson. But it's all caused by a change in membrane potential, so positive and negative charges. And if you remember, the sodium-potassium pump would pump out 3 sodium and pump in 2 potassium. So a positive charge on the outside of the cell is being created and a negative charge on the inside. So impulses along the nerve are caused by rapid changes in the membrane potential. Neurotransmitters are very key in this impulse because they have an effect on the sodium-potassium channels. So we're going to talk about the synaptic transmission process, where we have a neuron that's communicating with its target cell, so either a muscle or a gland across a synapse. So we can have an excitatory neurotransmitters that are going to cause the membrane potential, so the difference in charges between a membrane, to increase. Okay, so we see the increase here. So we look at a resting membrane potential as something that's the charge difference between the cell membrane, the inside and outside of the cell, when nothing's happening to the neuron. So it's just hanging out. It's just not doing anything. And excitatory neurotransmitters are going to cause it to become more positively charged. And we see this spike here. And we have acetylcholine, dopamine, and norepinephrine are neurotransmitters that have that effect. Similarly, but in the opposite direction, we have neurotransmitters that are going to make the membrane more permeable to potassium. So we have more potassium out of the cell and making the membrane potential more negative. And so that's where we see that dip in the resting, in the membrane potential here. So we go from a neutral resting value. And again, we don't have numbers here. We will see numbers in our next lesson when we're looking at this. But they dip down. As in any graph, it becomes more negative. You're going to see it go down. And we have GABA as a poster child for inhibitory neurotransmitters. And again, we see norepinephrine. So we see norepinephrine in both, and it depends on the target cell whether norepinephrine is going to be excitatory or inhibitory. And then we see this thing called a threshold. And what happens in order for a neuron to actually send its signal along the axon, it needs to have its value reach this threshold. So we need to have a bigger difference. The cell needs to become more and more positive on the inside in order for it to actually fire, if you will. And again, we'll learn about that a bit. That's called an action potential. So for it to actually release those neurotransmitters or make its effect on a target cell, it needs that action potential. So summation, as you can think of sum as it's the addition of things happening. So this is the effect produced by the accumulation of neurotransmitters from one or more neurons. And we're going to look at a couple different examples. Or just the accumulation of neurotransmitters from one neuron. So looking here, again, now we have numbers. And these are actually numbers that you would see. So typically the resting membrane potential is negative 60 or hovering around negative 70. And we see this is our postsynaptic neuron that we're looking at. And we have 3 presynaptic neurons that are making connections with D, the postsynaptic neuron. So we see A is going to cause a peak. So A is releasing excitatory neurons. But A alone isn't going to cause an action potential. B, again, excitatory, but no action potential. And C is an inhibitory. So C is like getting further away from an action potential. But if A and B are both sending the same excitatory messages, those two working together could cause a action potential to occur in D and make D send whatever message it is to its target cell. So we have 2 types of summation. We're going to look at temporal. So temporal is time, and we're going to talk about that in a second. And we have, in this case, here in yellow, we have our postsynaptic neuron. And so this is making a connection with its target cell. So again, a muscle cell or a gland. E is an excitatory neuron. E2 is also an excitatory neuron. And I is an inhibitory neuron. So that's what those are standing for. So there's 2 excitatory. If E alone sends a signal, an excitatory signal, so some neurotransmitters, it might cause a little bit of a raise in that potential. And again, looking at the regular, the average is resting potential is negative 70 for most neurons. And if E fires again, after a little bit, you're going to get about the same peak occurring. And in order for that postsynaptic neuron to actually do anything, it needs to have its peak reach this threshold here, the action potential threshold. But if E1, the same neuron, fires rapidly one after another, it could induce an action potential in the postsynaptic neuron. Okay? So sometimes a neuron just has to fire off more neurotransmitter more rapidly. That would cause a temporal summation. We also have spatial summation, where we have more than one neuron firing an excitatory, remember these are excitatory, signals to the postsynaptic neuron. So in this case, we have E1 and E2, so both excitatory neurons sending their neurotransmitters at the same time, and that is going to cause an action potential. However, if we have, for example, the excitatory and the inhibitory neurons, you're not going to have an action potential occurring. Okay, so we have the same neuron that can fire rapidly as a temporal summation, or we can have more than one excitatory neurons that are going to cause an action potential in the postsynaptic neuron.